Thank you very much, Dr. Emborg. And that's true grace under fire. So moving on to the next presentation by Scott Fahrenkrug from Recombinetics on the use of gene editing technology in cattle. Welcome.
Well, first off, no, we're not really modeling human disease in cattle. Although all the advanced reproductive technologies that are used in the fertility clinic largely came out of work in cattle, which, by the way, have 9 months of gestation, just like we do. But I'll tell you a little bit about recombinetics, and then I think at the end speak to some things about agricultural animals that have relevance, not only as models, but as food. Really pointing out here that the large proportion of our work in recombinetics is focused on developing spline models of human disease, but there is a portion of the company's effort that's trying to develop food faster, to identify superior performance genetics in livestock and to coalesce it into animals in a faster way than what's been previously undertaken. So I won't spend much time on this, but this is where I get on my soapbox and say, gosh, the CRISPR craze really is a craze. For the folks that are actually active in the gene editing field, there's been one player after another, and I've reviewed a number of papers where we've got yet better gene editing technologies, both in terms of their activity and their specificity, that are coming down the pipeline. I think we sort of generally started calling everything CRISPR at this point, but maybe there'll be a better acronym later. Not really sure. But essentially, there's the two editing platforms that really marked a difference in the efficiency with which these technologies can be deployed are pictured here. The tau effector nucleases and CRISPR, again, opening an opportunity for us to both inactivate genes and on the basis of homologous recombination to replace alleles with the alleles that we want. So again, just pointing that to published literature about some of the features of these technologies. I'll point out to you that in fact, talons can be used to multiplex. We've done up to 10 alleles at the same time using talons. We can do it with CRISPRs as well. It's very clear that the talons have so far the highest specificity of the editing platforms, although I haven't had the chance to work with the newest of the CRISPR enzymes. But how does one pick which platform to use? Well, what do you have running in your lab, and what can I order on the internet? It seems to be really what the deciding factor is. There's been a true democratization of gene editing when you can just order the components that you need to get the work done, as opposed to being a gene editor per se yourself. So just a few, I'll give a few examples of some of the products that we're developing at Recombinetics. First, Insurigen, the part of the company focused on disease models. I advocate we use the least expensive, most facile model you can bring to bear on a specific disease or physiology. So I got my PhD working on zebrafish, in Perry Hackett's lab at the University of Minnesota. So if I can do it in zebrafish, I'll do it in a zebrafish. But as has been mentioned, there's already some numerous examples where those organisms or even rodents are not good models of human disease, and in particular reference as preclinical models. Pigs have turned out to be a fantastic system, and I think most elegantly demonstrated by the importance of pigs in the cardiovascular disease research, both in the development of the surgical techniques and the devices that are brought to bear on treating patients. But really the similarities to humans are extensive. We generally had to rely on whatever spontaneous mutants would show up in pig populations, or some sort of dietary manipulation or surgical manipulation to get the phenotypes or the characteristics that we wanted to study. We'll say that there are some really valuable spontaneous mutations out there. In fact, one of the first pig models of cardiovascular disease was the so-called Rapaz pig, which results from a polymorphism, a hypomorphic mutation in the LDL receptor, where that version of that gene had about 60% of the normal LDL receptor activity for clearing cholesterol from the blood. So breeding for that allele resulted in really quite a fantastic model. It's got deficiencies, which is the justification for now making even better versions of atherosclerotic pigs. But by the way, you're eating those mutations. There's no selection. There's for elimination of those animals from herds. There's no extra danger that's been envisioned by anyone with regards to the use of those animals. And there's some other ones out there too. There's a mutation in the von Willebrand factor, one of the von Willebrand factors that caused a clotting disorder in pigs. Recently, there's been a couple of immunodeficiency models that result from spontaneous mutations picked up in breeding programs in pork production. And just really want to point out that the question came up earlier, what's the difference between a mutation and a variation? And it's being judgy. You call it a mutation when you're being judgmental. You call it a variation when you're not. And so I learned early on when I was at the USDA that we work with variations, which are just mutations. I'm just going to leave that there. We'll sort of come back to this point because, in fact, that mutation that resulted in that atherosclerosis model was a point mutation, a single letter that was different in the genome of the pig that made it have a predisposition to familial hypercholesterolemia. And we eat it all the time. In fact, count the number of pigs on the planet. There are 10 pigs with a mutation at every single nucleotide in the genome already walking around out there, already making their way onto our dinner plate. Now that makes a presumption, of course, that that allele wasn't deleterious to its survival or function, but then if we made that same allele in pigs using other methods, it wouldn't be viable or functional either. So we cannot invent or create a diversity of genetics that doesn't already actually exist out there. And to Carlisle's point, we always pick up what nature's already done and convert it to our utility. I was saying Angelica earlier was pointing out really what the rationale is behind using pigs as preclinical models, and that is really, we feel we can accelerate the development of therapeutic devices and drugs by using a more accurate model, whether that be with TALENs or CRISPRs, identifying the genetic predisposition in patients, and then replicating that genetic predisposition in pigs in a very precise way, changing as little as a single nucleic acid to reflect or to replicate the human congenital disease, and hopefully then providing a pig that serves not only as a safety model, which the FDA already strongly advocates for the development of therapeutics, but then to be also as functional as an efficacy model as well. And really the value proposition there has to do with how dysfunctional our drug development pipeline is. 91% failure of drugs going all the way from the beginning to the end of the clinical trials. That's at a cost of about $4 billion. That's for the bigger of the drug companies. Tufts did a study a couple years ago. It's about $2.5 billion per compound. And they're failing in preclinical trials. At the end of preclinical or in early clinical trials, these drugs fall out, and all for the absence of really a good, predictable preclinical model, which we believe pigs will provide. The concept here is use our pigs, fail early, fail often, and if you can't figure out the protocol in a large animal that has the same genetic problem that a patient does, then maybe you better move on to another compound or another protocol. Well, so this is just, again, one of those examples, the familial hypercholesterolemia model. The Rapaz pig was great, but it was only a a 60% loss of function of the LDL receptor gene. You had to use, these were farm pigs. You had to feed them cheeseburgers for 2 years, and then you'd get high cholesterol. You get atheroma formation. Generally, it's very focal in specific parts of the heart. We sought to develop pigs that had an atherosclerosis more consistent with human health or human disease. And so we ended up landing on this special little pig. This is a little mini pig called the Osaba swine. It has all 7 hallmarks of metabolic syndrome. This little pig population got dropped off an island off of Georgia, it's in the southern United States, and was left to evolve for 500 years by itself. It was feast or famine, its sole food source being just acorns. And so that's what ended up resulting, of course, is the insular dwarfism. That's a hallmark of these animals that makes them a better research animal. Since they're smaller, they max out at 80 kilograms, just like patients. But they have all 7 characteristics of metabolic syndrome. But still, you had to feed them cheeseburgers for a year before you could get the atherosclerosis. That adds up. So we went ahead and went in and started replicating some of the alleles for familial hypercholesterolemia as they'd been found in patients, putting specific mutations into these pigs. We've done it in a couple different ways. First, we've made a transgenic using the Sleeping Beauty transposon system for a gain-of-function mutant of PCSK9. Which is the subject of a new class of drugs. Several drug companies have recently gotten approval for these PCSK9 modifiers. And so not only does that trait affect human health, but these pigs may generally be a useful model for further testing of those PCSK9 targeted pharmaceuticals. The other was just like the familial hypercholesterolemia alleles segregating in patient populations. We replicated specific mutations in pig, which are a complete inactivation of the LDL receptor gene. In both cases, there are some subtle differences that I won't go into today, just like there were subtle differences in the patients and subtle differences in the mouse models that attempted to replicate this trait. The most important features are, though, that really at 3.5 months of age, we had clinically relevant levels of atherosclerosis and the beginning of the appearance of atheromas in all the right places in the pig. And this was our first endpoint study, up to 90% occlusion of the major vasculature. So here's a pig that's got diabetes and atherosclerosis, which we think will be really useful for the development of more effective devices, drug coating for stents, and even, as I said, pharmaceuticals. This is just another one of the slides, detailed slides of the histology of these animals, and just wanted to point out this one feature, another feature that makes these pigs really important. None of the pig models historically that have been presented have peripheral artery disease, but these pigs do. This is on regular pig food, so not feeding them cheeseburgers. They genetically must get atherosclerosis. And they get it in the periphery, which opens that up for the development of devices for dealing with peripheral artery disease, as well as the cerebrovascular devices that people are in the process of developing today. Well, the other point I wanted to make here was, unlike some of the studies I've seen studying atherosclerosis in pigs, you've got your experimental group, you've got your plus or minus high caloric diet, you've got your controlled group, and it was, people were generally, they had to feed those pigs high caloric diets for long periods of time, 2 years. We had people paying $20,000 for a pig, and only 30% of the time did they get an animal that actually had atherosclerosis, because of course there's complicating genetic factors as well that are contributing to their susceptibility. Every single one of these pigs got the disease, whether we fed them high caloric diet or not. We could accelerate it with a high caloric diet. And so in terms of reducing, having animals that give you that kind of reproducibility, I think is incredibly important. And having this ability to genetically edit pigs now opens up to make more effective models in a way that's essentially reflective of what has been done in basic biology for mice. So I'm going to whip through a couple more traits. Again, we don't try to develop models where there's other models that'll suffice, but this is a particular condition, the polycystic kidney disease, which is present in about 14 million patients in the US. The ultimate result being these patients lose their kidneys and they have to go on dialysis, and there was no model, zero. You make the mutations in mice, and they don't get it. So we made it in pigs, and this is just showing one of the more extreme alleles. This neonate was born with a kidney that looks a lot like a patient's, which is there are fluid-filled sacs in the kidney that compromise the function of the kidney, and they swell up to the size of footballs. This is an incredibly painful disease for the patients, and we only find out about it until it's too late. This is, I think, actually the most common congenital disease allele in the patient population. It's because we don't see it till later. This is an example of a trait we developed with the Mayo Clinic. They were actually looking for, they identified this mutation in a gene called RBM20 that results in dilated cardiomyopathy in their patients. So they'd have 40, 50-year-old guys show up at the clinic with an infarct, and turns out at least in that patient population they were looking at, this is largely due to a polymorphism in this RBM20. They wanted to develop models for stem cell therapy of dilated cardiomyopathy patients. And so the first thing we did was give them some neonates that were homozygous for this mutation. And without going too much into the detail of the slides, it was clear we had fibrosis in all the right parts of the heart. And at very high frequencies, every single animal has got the trait. And they've been using these neonatal pigs that we delivered to them as homozygotes as recipients of stem cell therapy. So it has some relevance even to neonatal health and therapy. But lucky for us, it actually turns out, of course, their patients came in as heterozygotes, and at 40 or 50 had an infarct. Lucky for us, at about 6 months of age, we start seeing some of the markers for cardiovascular disease and even heterozygotes. So both genotypes are going to be useful to us. And finally, another example of a model for which there wasn't a good model, neurofibromatosis. Geez, I remember when the mutation for neurofibromatosis 1 was discovered, and I was an undergrad or something, and still to this day, there hasn't been a good model, a sufficient model. It's a very painful childhood cancer that results in tumors right on the nerve bundles. So these little pigs have all the right characteristics of neurofibromatosis. They've got hypo and hyperpigmentation in the right places. They've actually got scoliosis, which is another characteristic. And we're now analyzing these animals with regard to their ability to process specific pharmacological agents that have been developed to deal with NF1. So really excited to have had this project funded by the Children's Tumor Foundation. I think there's numerous now cancer models in the lab that we've developed where pigs, it appears, are going to be better models than those that have existed out there. And so I thought I'd just give you an idea of the scale of pig model development. I know there's at least one other company in the audience and numerous academics who are all working to develop pig models of human disease. But they work. So far, we have failed to be let down. When we put the mutation that causes disease in a patient into pigs, they get the phenotype. Now, precise definition of the product specifications, no doubt the timing is not going to be perfect for everything. No doubt that the severity or the precise features of the disease are going to vary in pigs. But so far, we haven't been let down, and we see these as quite an effective approach to developing models of disease. But okay, I was asked to talk about cattle, so I'll do that. And in the context, I think, we'll come back to that point. Remember, we eat mutations every day. Every animal we cross has 3 million mutations that aren't present in any other animal. Every time we breed an animal together, 3 million new mutations. Well, given that, we still are able to select animals that have better performance characteristics than others. And we're focused in the company on all the same things that the livestock genetics companies are focused on. Better sustainability, more protein with less input, enhanced reproductive characteristics, reduced environmental impact. We have a class of products focused on animal welfare, and that refers both to disease resilience of the animals against some of the major pathogens that affect production. Also means fixing some of the congenital mutations that we've managed to select for in our identifying superior animals historically. The entire Holstein herd in the United States was built on the back of 8 bulls, whose semen has now been used to make daughters and granddaughters and all the combinations you can imagine. And so that resulted in, for example, in the Jersey breed, a mutation that's present in 37% of the animals that are on US soil. Homozygous results in a spontaneous abortion at about 60 days after pregnancy. So it really hits the bottom line of the producer because they've taken that animal offline to get them pregnant, and they lose it. So that's with 37% of your animals carrying the mutation. This happens way too frequently. Listen, if it was present at a low frequency, which has come up in the past, historically, one can just eliminate animals from future propagation. But to give up 37% of your genetic potential because you've managed to bring along this congenital disease allele is really, it's damaging. And of course, we can use gene editing to repair, to fix the allele and restore it to the one that we're all normally eating, the safe, the food that we've been eating for 10,000 years. So there's some opportunities in gender selection, but really we've been focusing a lot of attention on animal welfare, and this is, yes, of course, it's strategic to focus the discussion on the traits that really we feel we have a moral obligation to enable, but it's also just a fact that biologists love life. And we're working on these because we can, and we can solve problems today that have been facing the industry for a very long time. So just a few obvious things to go after. Now, I for the life of me, cannot figure out why someone has made double-muscled beagles, but it must have something to do with food. These are natural mutation, has 7 different times, 7 different breeds of cattle in Europe that were identified by ranchers because they saw an animal that had more muscle and they liked it. Hey, I'm going to win the state fair, or I'm going to be able to sell the service of my bull for more than the guy next to me because mine's bigger. Okay, so that resulted in the identification really of mutations all in the same gene, the myostatin gene, in different breeds, Belgian Blue, Piedmontese, Maine Anjou, Charolais. There's a second mutation in the Meiji-Anjou cattle, Marchigiana. All of these result in inactivation of myostatin to some degree, whether they be a point mutation or premature termination codon. They all result in the same thing. Animals, which, because they're not expressing myostatin, their muscle cells proliferate to a higher degree. So more muscle cells proliferating means more muscle, more meat, and actually lower fat. It results in a heterozygous condition of about 7% increase in retail product. As a homozygote, 30%. 30% more meat per animal. That's a huge number for somebody who's making pennies on each animal. Well, it comes with some deleterious effects as well because the fetus or the offspring or neonate is big. There are some dystocia problems, some delivery problems, and it's why the cattle that have these polymorphisms or variations are managed in highly integrated production systems Belgian blue cattle, you know, when they're making homozygous Belgian blue animals, they get cesarean sectioned. Okay. But there are ways to, if you have a homozygous bull, you can make all heterozygous animals. That's 7% more money. And there's really, it's already, the history has shown there's an economic model that makes sense for the use of this natural occurring variation. Well, we decided to use gene editing here. It's been quite some years now. Following this concept that you can take a superior cow, a superior bull, make an embryo, do an embryo injection. One can, in theory, do an embryo biopsy, and actually this is commonly being done now by genetics companies to identify embryos that have a higher genetic merit, put into a recipient, and out the other side comes animals that have either a high or a low genetic value based on luck of the draw, plus or minus your gene edit. And just like we always have in cattle, we cull the animals we don't want and we keep the ones we do. So we went ahead and deployed the technology, and this is a different breed of cattle. This is the Nelore cattle. It's the breed that's most important to beef production in Brazil. These animals are, basically Brazil built a beef industry in 50 years from scratch. They went and got these tough animals from India, which were disease resistant, thermotolerant, and just scrappy, tough guys. And they built a beef industry on the back of it. That's the largest, Brazil's the largest beef exporter in the world. It's ingredient beef. This isn't the highest quality beef. This is not your Ruth Chris Steakhouse beef. But what we did was just inject talons directed against the myostatin gene into embryos. About 75% of the embryos we injected had at least one allele modified. More than expected, we had homozygotes. And we put a couple of these guys on the ground, and this is just a picture showing, these are twins actually, because I didn't listen to my advanced reproductive physiology folks. I told them to put 2 embryos in every recipient. Yeah, well, I should have listened, but anyway, this one obviously is the edited one. The female next to this animal was not edited, so it turns out to be a pretty decent control. And this was a premature stop codon induced by a frame shift here that ultimately results in the identical protein being made as what's found in Belgian blue cattle. So we're now studying the segregation of these animals, and actually we're moving forward with these animals. They're actually mosaic. They've got 3 alleles. So out of the same experiment, we're getting controls, a novel mutation we made, and this new mutation here that looks like the Belgian blue allele. So we'll be characterizing the segregation of those alleles and the performance in those animals with regards to beef production. So another thing we went after, and it was, again, this has to do with animal welfare, is dehorning. There's about 5 million cattle a year that have their horns removed. It's not a very nice process. It's either liquid plumber on the head or a soldering iron to burn off the horn bud, or like bolt cutters, and then you reach in with a pair of pliers and pull the veins out and there's blood everywhere, and it's not nice for the animals. It's not nice for the people that have to do this, and everyone I've talked to that does this for a living has said, oh really, you can make that go away? Please do. So it turns out, of course, that there's a natural mutation that's present in some beef cattle that results in no horns. That polymorphism, that variation is present in incredibly low frequencies in dairy populations. It's likely to have, in fact, resulted from a contamination in the breeding program. The dairy animals that have the polled allele, the allele that means no horns, are generally at the bottom end of the dairy merit. This is a dominant trait. It could have been bred into dairy animals a long time ago, but you would take a huge hit on dairy performance. These animals, right, were started to be selected for differentially for beef and milk production 1,000 years ago. And so to cross a beef animal and a dairy animal, what you get out the other side is neither. And it would take you 8 to 10 generations of back crossing to recover the dairy merit if there had been an introgression using this traditional breeding approach. In fact, this would become the #1 sort of hit list or target for animal welfare advocates. I think Walmart and Nestle list this as their top focus in eliminating dehorning from the dairy industry. And so that really is a motivation for us to try to do something about it genetically. Well, back in 2012, I actually started trying to find the mutation. I joined forces with at least 4 groups in the world who were going after the mutation so that we had some strategies where we thought we might be able to identify the mutation and implement it in dairy animals. But back in 2012, a group of Medjugorje et al., one of the groups we didn't even know was working on this, discovered the causative mutations by sequencing animals across the population. And they identified 2 different mutations, each of which sprang into existence 500 to 1,000 years ago. One was pretty complicated. It's an 80 kilobase pair duplication and some polymorphism stretching across 260 kb, whereas the other mutation with the Celtic allele was a simple 212 base pair duplication into a 10 base deletion. So we sought to introgress that, and using this approach now instead, we have a superior bull. We can take cells and edit, identify the clones. This is back when I thought I'd have to screen through stacks of plates. Not. Very efficient. We can identify edited cells, and then one can clone and produce essentially the same animal, but now with the edit. Not going to go through all of the gels and the bands other than to tell you that depending on the experiment, we got introgression of from 1 to 5% of cells in culture that had, now this is more complicated. It's not a single nucleotide, right? This is both a deletion and an introgression at the same time, and it's a repeated sequence. So we're able to get that homozygous animals in 1 to 5%, which we thought was good enough to go ahead and proceed with making animals, and that's what happened. Not. Actually, that's what happened. Super cute, little calves. And turns out there's this side effect of having the polled locus. When you're homozygous, these little calves are born with an extra set of eyelashes, which just helps the cuteness factor, I believe. So we started this program in the company, and we were fortunate enough recently to receive funding from the USDA to support some of it and to do this into some additional animals. We're sequencing the genomes of these animals. We will have a report actually submitted in the next couple of weeks describing these animals. They now have been put on a truck and sent to California. They're now at University of California, Davis. Where Jim Murray and Allison Van Eneman are characterizing these animals with regards to their cow-like characteristics, and are they happy, healthy animals? And of course, they sure look like it. And so we're hopeful that a full characterization of these animals opens us up really to a commercial deployment of this technology. We could literally eliminate dehorning of dairy cattle, in this country at least, within a few years. If you were going to do this by natural breeding, even using marker-assisted selection, it would take you 50 to 100 years to get it done. So we can now actually make animal suffering go away. We didn't do this for fun. It's there to protect the animals from each other and to protect handlers from the animals. They don't need those horns anymore. It just seems like at 5 million animals a year, we really have a moral compulsion to make this go away. So I'm going to put out one other trait here, and that has to do with castration. Castration of pigs in the U.S. is, well, economically, it's pretty valuable. It's worth about $1 billion a year. But I'll tell you that worldwide, 500 million pigs have their testicles removed per year. It's not a very nice process. It's in the normal course of things. The people that are doing it don't have a problem with it, but the public does. In fact, castration of pigs has to go away by 2018 in Europe by legislation. Why do we do it? Well, we do it because you let the boars sexually mature and they become aggressive. And they taste like a locker room. It's called boar taint. It stinks. Nobody like, well, some Chinese like it, but pretty much the rest of the world does not like boar taint. So here we have a conundrum. The product we like, using a procedure we have been that causes animal pain, and a political environment that's pushing the elimination of that practice. Can we make it go away? And so this is an example of, this is now a more rational approach. We went and looked at the opportunity just to eliminate the pigs from sexually maturing, not go through puberty. And we went to the clinical data and found that there's, of course, a sexual differentiation pathway that's important across mammals. In particular, focused our attention on this GPR54 or KISS1. That's the KISS and KISS receptor system. Little kids would show up at the clinic and not, their testicles weren't dropping and they weren't sexually maturing. And so the doctors figured out, well, if I give some hormone to this kid downstream, GnRH or FSH, then they'll go through the sexual differentiation and they'll be fine. So we wondered if we went after that, what would happen to the pigs, and it's kind of embarrassing, but their testicles don't drop. In fact, they, at 6 to 12-month-old pigs, the testicles are not well developed. There's no boar taint. They act like barrows, like they have been castrated. And we think this represents really a good concept for eliminating that procedure. And it's rescuable. So yeah, it seems somehow less objectionable to show you the testicles off the animal. Essentially, we can take in with hormone injections into the animals, we can actually, as opposed to this is now the knockout pig, its testicle didn't fully develop, we can hit it either with GnRH or FSH and they will sexually develop. So there's a way to actually reproduce these animals for production, but in the terminal cross, no testicles. So one of the things that's been really striking to us about gene editing is the ease with which we can situate it within what the already existing production systems are, genetics and production systems. Really, we can accelerate genetic improvement either in somatic cell, by somatic cell nuclear transfer. We've used it in IVF embryos. We've actually used it in semen-mediated delivery of editors. And of course, we can identify young stock that has a superior production characteristics that we can immediately clone and make animals with the edits that we want. And then I'll leave you with what is really probably what I think is the future of gene editing and livestock genetics, and that is that we're just talking about Mendelian traits right now. We've done 10 alleles at the same time. There's no doubt in my mind we'll be doing 50 or even 100 alleles at the same time. And that has real value to our ongoing genetic selection regimens that are underway in livestock around the world. And this is a little study done by John Hickey at the Roslin Institute, which shows here's the normal genetic improvement profile just based on whole genome selection. And then this is what happens when you have either 5 or 10 medium effect alleles that are present at a low frequency in a population introgressed. And what you can see is really a dramatic increase, a dramatic jump in the quantitative performance of animals were one to put in these, introgress these production alleles. So we can use this we talked about precision crossbreed. As we're doing whole genome selection on all our populations, we have at least an ability to resolve our bad moves, go in and fix those congenital mutations. We can break linkage disequilibrium. We've got positive and negative alleles very close to each other in the genome that will never segregate away from each other because we don't have enough meioses like plants do, we can actually separate them using gene editing. And of course, there's an opportunity for novel alleles in the area of disease resistance and resilience, which is really the #1 killer of animals, at least until harvest. And then I've been told to always put up what you want. This is what I want. We think that those alleles that are already out there that we've been eating for 1,000 years, whether we introgress them by editing or introgress them by breeding, should not be regulated in the pre-market mechanisms that have been used so far. And that if there are some novel alleles, that would justify pre-market regulation. But really, we are talking about taking advantage of biology, of nature's bounty, and we hope we can use gene editing to bring some of those solutions, and we can get regulation in a rational world. With that, I'll take any questions.